See you guys. Yeah.
Yeah, it's hard when you get
Good morning. Welcome to another Wednesday. A reminder, Friday is test 4. Test 4 is almost written. Test 4 is only going to cover the material that was on Monday's lecture and today's lecture is going to be relatively short, I think. Okay, so there's not a lot of material, so hopefully you guys will do well. Not a lot of things to study. There's 20 questions, and as it says up there at the moment, there's only one multiple. I know you guys hate these multiple, so in principle, everyone should do well. I'm having an extra Zoom office hour tomorrow. So tomorrow at 10 o'clock to 11 o'clock, there's an extra Zoom. There's an office hour this afternoon, and then there's one tomorrow. I will send out an announcement this evening about the office hour and to remind you guys about the test for Friday. Okay, so Friday will be test 4, it'll be the last test. So for those of you who do not write 3 tests, so if you've missed 2 or more tests, you can write the makeup if you have the right documentation. So if you've missed 2 or more tests and you have documentation, there'll be a makeup test next Wednesday, not next Wednesday, next week. I'm not sure when next week, depending on the availability of the students that have to write it. So after the test Friday, I will email people that have missed more than 2 tests, and you will be writing the makeup. All right? So, when last we met, we were talking about cell adhesion molecules and various aspects of cell adhesion and function. So we talked about tight junctions and gap junctions, and then we talked about these cell adhesion molecules that are involved in cell-cell adhesion, which are homophilic, or cell and other sort of adhesion that are heterophilic. So if you're homophilic, we know it's cell-cell. If you're heterophilic, you don't know what it is, it's just something different. And we talked about specific examples using cadherins in this IG superfamily, and then we talked about integrins and how they bind to the ECM. And then we started talking about selectins. And when we're talking about selectins, which bind to carbohydrates, we use the example of extravasation. So extravasation involves a number of these cell adhesion processes. Extravasation is when a immune cell, a leukocyte, leaves the bloodstream to have to cross this endothelial cell layer. This endothelial cell layer is held together by cell adhesion molecules. It probably has tight junctions in it. One of these cell adhesion molecules is a molecule called ICAM. ICAM is part of the IG superfamily. So in between these cells is cell adhesion molecules, including ICAM. That ICAM can also be used for other adhesions. So ICAM is involved in cell-cell adhesion, but it's also going to be involved in this adhesion with integrins in a little bit during this process. So I've talked about this process last time. I'm going to show you a movie, and I'm going to do it again. So let's see if this is going to work. So this movie is in your Achieve. So this is from Achieve, as I'll show you in a second. So it's in Achieve in my lecture resources in week 8. Okay, so you have access to this movie. Let's see if this movie works. Movie works, just the audio doesn't.
In the blood, in the text.
let me back up.
The presence of an infection or inflammation triggers leukocytes to move into tissue.
Let's do that again.
Leukocytes, or white blood cells, normally circulate in the blood unattached. The presence of an infection or inflammation triggers leukocytes to move into tissue toward the foreign invader in a process called extravasation. Extravasation requires a succession of specific cell-cell contacts between leukocytes and endothelial cells lining the blood vessel. First, inflammatory signals induce endothelial cells to exocytose P-selectin and synthesize platelet activating factor, PAF, both of which appear on the cell surface. P-selectin molecules bind carbohydrate groups at the leukocyte cell surface. This weak binding to selectin, coupled with the force exerted by the flowing blood, causes the leukocytes to roll. The leukocyte also contains PAF receptors and integrins. Receptor binding to PAF results in the activation of the integrins. The activated integrins are able to bind ICAM molecules constitutively expressed on the surface of endothelial cells. This causes the leukocyte to stop rolling and to tightly adhere to the endothelium. The adhered leukocyte then begins its migration between adjacent endothelial cells to reach the underlying tissue.
All right, so that is part of Achieve, and I'll talk about that in a second again. Okay, so this movie is in Achieve in my resources, lecture resources under week 8. Okay, so you can find it there. So, it's all about changing cell adhesion. Normally, endothelial cells do not want to bind to blood cells. Here, you want the endothelial cells to bind to the leukocyte. So the endothelial cell has to change. So in response, I'm not showing you anything, am I? My apologies. So the endothelial cell has to change. So normally, nothing is binding to these endothelial cells. It's going to be selective, and it's going to only pick leukocytes. And it does that by secreting P-selectin. So in the absence of inflammatory signals, there is no P-selectin. In the presence of inflammatory signals, there is P-selectin. It's also secreting this PAF, but that's not involved in cell adhesion, that's just a ligand. Okay, P-selectin is a cell adhesion molecule that binds to carbohydrates. Those carbohydrates are found on the leukocyte. So the leukocyte will bind to P-selectin, it will slow down. When it slows down, PAF can bind to the PAF receptor on the leukocyte. When PAF binds to the PAF receptor, it activates integrins that are on the leukocyte. These integrins can then bind to their ligand. It just happens to be this cell adhesion molecule called ICAM. Okay, ICAM is found on these endothelial cells. It's doing its job in endothelial cells, but this ICAM happens to have an RGD sequence in it that's recognized by this integrin that's on the leukocyte. Okay, so while the ICAM is undergoing this homophilic interaction, the leukocyte integrin is going to undergo a heterophilic interaction. So you have heterophilic integrin binding to this ICAM molecule. And that binding is strong. That binding then adheres this leukocyte tightly to the endothelial cells, and then it allows it to extravasate, which is pass between these endothelial cells without really doing any damage. It's regulated, and it can go out and fight the infection. Okay, so this is extravasation. It's an example of how P-selectin, which is a cell adhesion molecule that binds carbohydrates, functions, but it's also showing you how integrin functions, and you also have the Ig superfamily with the ICAM molecule in their function. So it kind of shows you a whole bunch of things. Okay, so that's sort of the end of cell adhesion and sort of the cytoskeleton stuff, and now we kind of roll into membrane and membrane proteins. All right, so there is hopefully eventually going to be a tutorial more specifically talking about lipids and membranes. Okay, so there will be a tutorial on lipids and membranes. The tutorial information is not on the final exam. Okay, I keep getting emails, I'm not sure how many times I have to repeat it. The tutorial information is not on the final exam. You'll be tested on a quiz for the tutorials. Okay, so the tutorial information is separate than what I'm going to be teaching you. And so for the final exam, you need to know it to this level. All right, so we're going to talk a little bit about membranes. In the tutorial, there'll be a lot more about membranes, but that will not be on the final exam. All right, so membranes, biomembranes. Okay, so the membrane is sort of what defines the cell. So there's a lot of words on this slide, almost none of it testable, just kind of giving an overview. The membrane is defining the cell. The membrane is made of these amphipathic molecules, these phospholipids and other molecules that are amphipathic. So when you're amphipathic, you have a hydrophobic and hydrophilic domain. If you're in a hydrophilic environment, you try to hide that hydrophobic domain. So biomembrane is formed because they're trying to hide these fatty acid chains that are hydrophobic. So these phospholipids and other sort of components that make the biomembranes are hiding the fatty acid chains, and they normally do it through making these membrane structures. There's other structures you can make using these fatty acids. There's things called micelles and liposomes, which are smaller structures. But basically with all of them, you are hiding these hydrophobic domains, and when you do that, you form this structure that actually acts as a barrier. Right, so now you have this structure, which is this biomembrane, this lipid layer that's a barrier. You can put things in that barrier and give it functions, and that's exactly what happens. Cell membranes have proteins in them, and while cell membranes have a little bit of function on their own, most of the functions of the membrane is because there are hundreds, if not thousands, of different proteins that can be embedded in the membrane. So the proteins in the membrane are really what's giving a lot of the function. And we'll talk about the different types of proteins you can put in the membrane for the rest of this course, or the rest of my section at least. Okay, so biomembranes, so these are the membranes that are surrounding cells. Okay, so they are functional because of certain properties, and all biomembranes have these 4 properties that we're going to go individually and describe individually. 4 properties, almost a multiple choice question that writes itself. Okay, so a biomembrane is fluid, it's a closed compartment, it's semi-permeable, and it's asymmetric. So let's go through one of these one at a time. Okay, it's fluid. So things move rapidly in 2 dimensions. So when you look at the biomembrane, it's a phospholipid bilayer. It has 2 leaflets. It has sort of an outer leaflet and an inner leaflet. So there's phospholipids on one side and phospholipids on the other side, and they're not necessarily the same types of phospholipids, right? These phospholipids and other things in the membrane can laterally diffuse. So there is rapid lateral diffusion. Things move laterally in the membrane all the time, and that's important to the function of most membranes. So these lipids and things inside the lipids laterally diffuse. There's very rare movement from one side to the other. So if you're sort of on the cytosolic side, you rarely move to the exoplasmic side, to the other side. So there's movement within the leaflets, within each half of the lipid bilayer, but there's very little movement between the two, between the inner and the outer leaflets. That fluidity can be altered. Different cells have different fluidity in their membranes. That fluidity is determined by a number of factors. One of them is sort of the makeup of the fatty acids that's found in the phospholipid bilayer. Okay, there's going to be more details on this, and I'm sure you know this from biochemistry and other courses. If you have longer fatty acid chains, you're going to have an increase in the melting temperature. If you put in double bonds, so if you have sort of different double bonds, you're going to have a decrease in the melting temperature because you're adding kinks into it. So you don't have to memorize the specifics for the exam because it's going to be on the tutorial side of things, but appreciate you can change the fluidity of the membrane by changing the length of the fatty acid chains and by changing the number of double bonds. So double bonds and length of chains do affect fluidity. You can also affect fluidity by adding sterols into the lipid bilayer, things such as cholesterol. Again, more details will be in the tutorials, but for our purposes, sterols affect fluidity. Proteins also affect fluidity. If you put a protein inside the membrane, you can then anchor that protein to something. So for example, if you have a transmembrane protein and it's linked to the cytoskeleton, that transmembrane protein is not going to move, therefore the lipids around it are less likely to move. So proteins can affect stability, or fluidity I should say, because the proteins can be anchored, and if you have a lot of anchored proteins, it's going to restrict fluidity. Temperatures can also affect fluidity, however, that's not necessarily a biological mechanism that we use. Our cells have different fluidities, different cells have membranes that have different fluidities, but it's not because they're different temperatures. However, it's important to recognize that if you lower the temperature, there's going to be less fluidity, of course. So experimentally, that's an important tool. Okay? Fluidity can be measured in a number of ways. One is a process called FRAP, which is fluorescence recovery after photobleaching. Okay? So FRAP, fluorescent recovery after photobleaching. There will be more details in the tutorial, however, I'm going to give you some details here. So this is basically measuring the fluidity of proteins that are in membranes. So if I back up a bit, there are lots of proteins that are present in membranes, and these proteins are not tethered to the cytoskeleton or the ECM. Those proteins can laterally move inside the plasma membrane. So if you look at proteins moving within the plasma membrane, the fluidity of it, it's sort of a measure of the fluidity of that membrane. So FRAP takes an advantage of that. All cells have lots of proteins within their membranes. You can take those proteins and you can label all of the proteins. So you do an experiment where you've labeled all of the proteins on the cell surface of a membrane fluorescently. So now they're all fluorescent. So when you look down on a cell, you can measure the fluorescence. So you have a cell, all the proteins in its membrane are fluorescent, and you measure the fluorescence. You can look down at the microscope and you measure some sort of relative fluorescence, and you give that fluorescence a number. Here it's, whatever this is, 2,900, you can imagine it's 100%. So there's 100% fluorescence, that's your baseline. You then photobleach. What does that mean? You take a really bright light and you zap it on this fluorescent protein, and you're basically bleaching it so it can no longer be fluorescent. So photobleaching bleaches an area so it has no fluorescence. And then you measure how much fluorescence there is in that area, and you will have a decrease in fluorescence after bleaching. Right? So that will be your other level. So you went from 100% to something like 10%, let's say. You then wait for recovery. It's fluorescence recovery after photobleaching, and you wait. What are you waiting for? You're waiting for proteins to move into this area. So this area has no fluorescent proteins. Around it, the rest of the cell has fluorescent proteins. If there is fluidity, some of the bleached proteins will move out of that area, and some unbleached proteins will move into that area and cause fluorescence. So if there is fluidity, you will get a recovery of fluorescence. And that's what you're measuring over time. So in this experiment, for example, before photobleaching, you have a certain level of fluorescence. After photobleaching, you have a reduced number of the level of fluorescence. And then you measure over time, and you get sort of this new state where you see how much of the fluorescence is recovered. Right? So in this example, 50% of the fluorescence was recovered, 50% wasn't. So that means that half of the proteins are basically attached. So this gives you a little measure of fluidity of that membrane. If you do this with a different cell, you're likely to get different numbers because different cells have different fluidities. Okay? If you try to do this with a naked phospholipid bilayer, if you had a way to do it, you'll see that in general diffusion is quite quick, but here it's much slower because there's proteins restricting movement, and a lot of these proteins are bound to other proteins, such as proteins in the cytoskeleton, and therefore they don't move or they move much, much slower. Make sense? So that's sort of the example. This is a movie that sort of shows that too. So there's going to be two movies. One where they're going to bleach an area and there's going to be a recovery, and another one where they're going to bleach an area and there's going to be no recovery, to show you the different membranes have different fluidities. So that's how this works.
Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
Okay, so you make membranes fluorescent, you measure fluorescence before bleaching, you measure fluorescence afterwards, and you look for recovery. If there's recovery, it means that there's lateral diffusion. If there isn't recovery, it means that there is no lateral diffusion. So, first example, in the ER, there's lots of movement in the ER. In this example here, it's lamins within the nuclear lamina. They don't move, so there is no recovery. All right? So, biomembranes are fluid. Biomembranes are a closed compartment. Okay, they are a closed compartment that maintain their symmetry, which is sort of the last thing we're going to talk about. Okay, so by closed compartment, when you follow the membrane, you can see that there's always an ectoplasmic and a cytosolic face. So it's a bilayer. There's always half of the bilayer is always ectoplasmic, and the other side is always cytosolic. So exoplasmic is the same as being extracellular. And cytosolic is obviously it's always facing the cytosol. So when you look at endocytosis or exocytosis or whatever was going on, you can consider any lumen basically the same as being outside. So if you're in the ER, if you're in the Golgi, if you're in a lysosome, you are basically the same as being outside of the cell. You are luminal, which is basically being almost like extracellular. Here the term is exoplasmic. Okay, so the exoplasmic membrane is always facing the outside, either the outside of the cell or the lumen, which is basically the same as being the outside. If you are facing the cytosolic side, so if you are the half of the lipid bilayer that's facing the cytosolic side, you're always facing the cytosolic side. So when you're forming different compartments within the cell, those compartments always have that same orientation. It has cytosolic side, and it has an exoplasmic side or extracellular side, and that is maintained. So when you put a membrane, when you put a protein in a membrane, you put a transmembrane protein in a membrane, that protein always has that orientation. So if you have a membrane protein in here, and it has an extracellular domain and a cytosolic domain, that is always maintained. That domain is always extracellular, that cytoplasmic domain is always cytoplasmic. That's true when it was in the ER, it's true when it's in the Golgi, it's true when it's in the secretory vesicle, it's true when it's in the membrane, the plasma membrane. That asymmetry, that closed compartment is maintained under all states. So if this is cadherin as an extracellular domain and a cytoplasmic domain, that extracellular domain is always extracellular. It's always in the lumen of the ER, it's always in the Golgi, and eventually it's on the cell surface. Cytoplasmic domain that can interact with the cytoskeleton is always going to be in the cytoskeleton. It's going to be always in the cytoplasm. Okay, so membranes are fluid, membranes form closed compartments, membranes are semi-permeable. I think you guys have done this at many different levels, using many different examples. We're going to keep it simple for test purposes. If it's small and uncharged, it can pass through the membrane. Small and uncharged, it can pass through the membrane. If it's large and hydrophobic, it will not pass through the membrane. So if you're large and hydrophobic, you have to be transported. And we'll talk about membrane transport next week. So small uncharged goes through, can diffuse across. Large and did I say hydrophobic? Yeah. Hydrophilic, my apologies. You guys can read. And you should know what it is anyway. If it's large and hydrophilic, it cannot cross. Right? Because there's hydrophilic molecules both outside of the cell and in the cytoplasm, and the phospholipid bilayer has this hydrophobic domain in the middle. So large and hydrophilic will not cross. It has to be transported. Right? So semi-permeable, and the last one, which you already sort of described, it's asymmetric. The membrane is asymmetric, and that symmetry is always maintained. Right? The outer leaflet, the exoplasmic, is different than the inner leaflet, the cytosolic leaflet. It's got different phospholipids in it. But additionally, the proteins that are present within this membrane are asymmetric. They have a certain orientation, and that orientation is always maintained. If a certain domain is always going to be cytosolic, or a certain domain always going to be exoplasmic or extracellular, it's always that way. Cadherin is always in the membrane that way. It's always asymmetric as a cytosolic extracellular domain. Points like that should be brought home in situations like this, so you don't have to memorize any of this. So if you consider sort of ATP synthesis or how the chloroplasts work in terms of generating ATP as well. So whether you're generating ATP in mitochondrial membranes or ATP in chloroplast membranes, there's a whole bunch of proteins that are pumping things and moving things around. The orientation of all of these membrane components has to be this orientation, this asymmetry. You can't flip a proton pump or something sometimes. It always has to be that orientation. It always has to have that asymmetry to it. Okay? So the components of the phospholipid bilayer are asymmetric. The proteins in the bilayer are asymmetric, always. And always carbohydrates are found on the exoplasmic face. So cell surface proteins, things such as cadherin, the extracellular domains are almost always glycosylated and have carbohydrates on them. Okay, so glycosylation is a standard thing that happens to extracellular domains of proteins. That kind of adds to their asymmetry. Okay, so now we kind of move into transmembrane proteins and this asymmetry that I was talking about. So the membrane itself has these four properties of whatever I just said. There's fluid, semi-permeable, closed compartments, and asymmetric. And within that asymmetry is proteins. Proteins that are in membranes are also asymmetric. And there's three types of proteins that can be in membranes. As I said, all of them are asymmetric. So those diagrams like this are kind of helpful, but they're kind of also confusing because you can't really tell what the heck is going on. Right? So, but I think you can see some membranes span some proteins span the membrane. So this protein is spanning from one side of the phospholipid bilayer to the other side. So that's an integral membrane protein. It's integral, it's part of the membrane. Some of them are lipid linked. They do not go through the membrane, but they are still linked to the membrane. Other ones are peripheral. They are not directly attached to the membrane, but they are attached to something that is attached to the membrane. And we'll go through each one of these individually. Okay, so there's three types of proteins: integral, lipid linked, and peripheral. Starting with integral membrane proteins. So we've seen a bunch of these proteins already, cadherins and integrins and a whole bunch of other receptors that we might have talked about or Rob might have talked about. All integral membrane proteins have three domains. They have a cytoplasmic domain, a transmembrane domain, and an extracellular domain. Okay, let's start off with the transmembrane domain, because that's sort of the easiest one to kind of picture to some extent. The transmembrane domain is going to span the lipid bilayer. That lipid bilayer has these hydrophobic tails. So the transmembrane domain has to be hydrophobic. So transmembrane domains are made of hydrophobic amino acids, and those hydrophobic amino acids are spanning that hydrophobic region of the lipid bilayer. So those hydrophobic amino acids develop, or they use certain conformations. So those hydrophobic amino acids either form an alpha helix or a beta barrel. You don't have to memorize what these things look like, but these structures are secondary structures that amino acids form under certain situations. Here, these hydrophobic amino acids are forming this structure called an alpha helix or a beta barrel, and those hydrophobic amino acids are the ones that are going to be inside the phospholipid bilayer. So you need enough hydrophobic amino acids to span that bilayer, and that's usually, in the case of an alpha helix, about 20 or 25 amino acids is what makes up the alpha helix that's going to span that phospholipid bilayer. Okay, so you have a transmembrane domain. You have an extracellular domain. So part of the protein is extracellular. As I already mentioned, that extracellular domain is often glycosylated. Almost all proteins that are transmembrane proteins on the cell surface are glycosylated on the extracellular domain. So the extracellular domain is probably already hydrophilic. Once you glycosylate it, add carbohydrates to it, it's going to be even more hydrophilic. So you have an extracellular domain that's very hydrophilic because you just glycosylated. You have a transmembrane domain that's the opposite, is hydrophobic. And then you have a cytoplasmic domain that's also going to be hydrophilic. That cytoplasmic domain often has these polar groups, these charged groups that are close to the polar head groups of the phospholipid bilayer. So the phospholipid bilayer has these hydrophilic groups that are polar, and you have these charged amino acids on the cytoplasmic domain. So these charged amino acids are interacting with the charged molecules on the phospholipid bilayer, but more importantly, these charged amino acids, so here you have these blue amino acids, these charged amino acids are going to not want to go into this phospholipid bilayer. So if you pull on this hydrophobic protein, if you yank on this extracellular domain and you try to pull this protein through, these charged amino acids will fight that because they do not want to go into this hydrophobic region. So here you usually have arginine or lysine present. So if you're anchoring, so if this is a cadherin and you're sticking two cells together, you don't want the cadherin to slip through the membrane, right? And it doesn't because you have these charged amino acids. These charged amino acids do not want to go into this phospholipid bilayer because it's hydrophobic. Similarly, you have these huge domains that are charged on the outside. You have all these glycosylated structures, so you can't pull the protein into the cell either. So you're kind of anchoring it in place because you have charges on one side of the membrane, charges on the other side of the membrane, and the middle is hydrophobic. So hydrophobic amino acids don't want to go in a hydrophilic area, and all these charged structures on both sides don't want to go into the hydrophobic area. So you've anchored this protein quite well. A lot of rambling, but hopefully makes sense. Okay, so you have integral membrane proteins, you have lipid-linked proteins. So what are these? These are proteins that are linked directly to existing structures within the phospholipid bilayer. So there's different phospholipids, glycolipids, sphingolipids that are present in the phospholipid bilayer. You can link proteins directly to them. So you can link proteins to the exoplasmic leaflet, or you can link proteins to the cytosolic leaflet. The way you link proteins to the exoplasmic leaflet, so these are all these extracellular proteins, is through something called the glycosyl phosphatidylinositol anchor. GPI anchor. I didn't name it. Okay, so GPI anchor is always extracellular. Okay, so phosphatidylinositol is the phospholipid that it's bound to, but you don't need to memorize it. You don't need to memorize the name. Just recognize it. GPI anchor is extracellular. It involves sugars, but the protein is always going to be extracellular. We'll talk about how you form a GPI anchor next week, but for this testable thing, GPI anchor always extracellular. Okay, and linked directly to the phospholipid bilayer. You could also link proteins directly to the inner leaflet of the bilayer through processes called acylation or prenylation. So if you have a protein that's in the cytosol, if it has a N-terminal glycine, you can undergo this chemical reaction called acylation and link a protein, this glycine, right to an existing structure within the lipid bilayer. Okay, so you have the inner leaflet of the bilayer, you have certain structures within that. You can link proteins through acylation, more specifically N-terminal glycine. So proteins are linked to N-terminal glycine to the phospholipid bilayer through a process called acylation. You can also prenylate. Prenylation is when you link, this time it's a cysteine that's close to the carboxy terminal domain or at the carboxy terminal domain. So prenylation is a protein that you're linking through the carboxy-terminal domain. Acylation is a protein that you're linking through the N-terminal domain. Okay? With prenylation, it's a cysteine residue at or near the carboxy-terminal domain. We've already talked about a prenylated protein. I'd like to remember that. It's lamin B. Lamin B is prenylated to the inner nuclear membrane, and now you know what it means. It's directly linked because lamin B has a cysteine residue that's at or near its carboxy-terminal domain that allows it to hold the inner layer of the nuclear membrane in place. All right. So with all of these lipid-linked proteins, the protein does not enter within the phospholipid bilayer. The protein is not in the bilayer, unlike the integral membrane protein. These proteins are sort of linked to the membrane. Because they're just linked to the membrane, and because these phospholipids and other structures can laterally move, these proteins, lipid-linked proteins, tend to have very rapid lateral diffusion. So these proteins can move around very quickly. Easy to understand, I hope. All right, so we have integral membrane proteins that have extracellular, cytoplasmic, and cytosolic, extracellular, transmembrane, and cytosolic domains. You have lipid-linked proteins that can be GPI linked on the outside, or prenylated or acylated on the inside of the membrane. You then have something called peripheral membrane proteins, which is sort of a catch-all. All a peripheral membrane protein is a protein that's either bound to a lipid-linked protein or a transmembrane protein. So you need an existing lipid-linked protein or a transmembrane protein, and the peripheral membrane protein will attach to it non-covalently through things such as protein-protein interactions or charge interactions. So again, when you kind of look at schematics like this, you have to have existing transmembrane proteins or lipid-linked proteins, and then you can link something else to it, and that's a peripheral membrane protein. Okay, these peripheral membrane proteins are also asymmetric. If it's a cytosolic peripheral membrane protein, it's always in the cytoplasm. If it's something extracellular, it's always extracellular. We've sort of talked about some of these proteins already, particularly with linking things to the cytoskeleton. We know that catirins or integrins or Ig superfamily molecules link to the cytoskeleton, and we've already mentioned adapter proteins. Well, those adapter proteins are basically peripheral membrane proteins. You can have a cationin that's linked to the cytoskeleton through these peripheral membrane proteins, or an integrin bound to the cytoskeleton through these peripheral membrane proteins. Okay, so peripheral membrane proteins are linking already proteins that already exist in the plasma membrane, either transmembrane proteins or lipid-linked proteins, to other proteins either on the outside of the cell, to the cytoskeleton, or on the outside of the cell, to the extracellular matrix. Okay, so we have 4 functions of membranes, 3 types of proteins. Okay, we will next talk about how you get those proteins in the membrane, but not on this test. So run and be free. Good luck Friday. Yeah. See you Monday. Okay, I still have mine from this morning. Thanks.
I think that was under 10.